Hello everyone. Hi, my name is Priyal Bagwe and I am a second year graduate student in Mercer University College of Pharmacy. Today, I'll be talking about my project titled Exploring the Potential of Bioengineered Cell-Based Therapy for Parkinson's Disease. Parkinson's disease is the second most common neurodegenerative disorder associated with decreased dopamine levels in the brain. Parkinson's disease affects more than 1% of population over 60 years of age and about 4 to 5% of the population above 85 years of old or older. The current treatments for Parkinson's disease are all symptomatic relief. The goal of our study was to assess the potential of a regenerative medicine based cell therapy approach to increase the dopamine levels in the brain. In this study, we used rat adrenal pheochromocytoma PC12 cells that can produce, store, and secrete dopamine. Our strategy is to encapsulate these PC12 cells in the microcapsules. The microcapsules have a selectively permeable polymer membrane that will allow the entry and exit of only necessary molecules. For example, it will allow entry of glucose, oxygen, and nutrients and exit of released dopamine and will protect the cells from immune responses. We formulated the microcapsules and analyzed them. In terms of the formulation of microcapsules, minor modifications were made to the Buki Mini Spray Dryer Bean 190 to formulate the microcapsules encapsulating the PC12 cells in a continuous process. The parameters that affect the size of the microcapsules, that is, the flow rate, nozzle diameter, pump speed, concentration of sodium alginate solution, were all varied to optimize the size of the microcapsules. We further characterized the microcapsules for their size at different pump speeds, different flow rates, and different concentration of sodium alginate solutions. We found that the microcapsules were relatively reproducible and scalable, and thus can be made in larger quantities as a continuous process with narrow size distribution. The FTIR spectra of the microcapsules confirmed the formation of calcium alginate in the microcapsule membrane, and this was confirmed by reduction in the peak intensities of free carboxyl groups at 1593 and 1406 centimeter inverse. We further analyzed the stability of microcapsules. The influence of contact time between the chitosan glutamate solution and the cross-linked alginate microcapsules was evaluated, and it was found that the microcapsules were stable throughout the experimental duration. Selective permeability of the microcapsules was evaluated by quantifying the dopamine release from the PC12 cells from the microcapsules and the protein content inside the microcapsules. The protein permeability of alginate chitosan membrane was determined to ensure that proteins do not cross the membrane. Nitric oxide is an important marker for innate immune response. The antigen presenting cells like dendritic cells and macrophages were analyzed for the release of nitric oxide upon its exposure to an antigen or a foreign invading pathogen. The microcapsules were non-immunogenic. Thus, we successfully formulated microcapsules and they were found to be stable for a period of 30 days and were selectively permeable and non-immunogenic. Taken together, This can serve as a potential alternative or an adjunct treatment strategy for Parkinson's disease. Thank you, Georgia Bioinnovation Summit, for giving us this opportunity to present our research. Thank you.